Hello, everyone, and thanks for joining us for the Innovations in Disease Modelling 2023 online symposium. I'm Caroline Clark, conference producer for Technology Networks, and I'm here to moderate today's event alongside my colleague, George Hammond. Before we start the talks today, I just have some general information and a few tips on getting the most out of the event. Firstly, as well as the fantastic lineup of speakers that we have for you, I'd like to point out the networking areas on the platform, which you can use to chat to other delegates throughout the day. On the left hand side, you will see the People tab, where you can access a list of attendees that you can contact. From here, you can message them privately, and all your ongoing conversations will appear in the chats area. I encourage you to get involved and take this opportunity to speak to some of the over 1,000 plus delegates that have registered for the event. If you are not sure who you would like to chat to, we encourage you to use our participant matching feature to match with attendees who share similar interests to you. Just navigate to the participant matching located in the left hand menu and fill out a brief questionnaire. Our software will then do all the hard work for you and match you with other attendees with similar interests and goals. In the list of matches, there will also be a percentage match, which indicates how close of a match your responses were to the attendees in the list. Please remember that we encourage debate and discussion, and we welcome all of you to use our networking services to introduce yourselves to other attendees. But we do not permit the overt solicitation of products or services and reserve the right to block any user who infringes on this rule. On a similar note, all of today's talks offer the ability for you to engage with our speakers via the Q&A session at the end of each talk. I encourage all of you to ask questions, as this is the best opportunity to get the live answers from our panel of experts. We will explain how you can ask questions at the end and start of each presentation. It's also incredibly important to say that this event was only made possible thanks to the generosity of our Technology Spotlight sponsors, BitBio and Biotechne. In the Literature and Resources section, you can download free educational pieces from our sponsors and partners, including application notes, white papers, and guides focused on disease modelling. For more detailed instructions on how to make the most of the features available to you at this event, please navigate to the event homepage and watch the overview video at the top of the page. We would like to ask if you could refrain from video, audio, or photo recording of all talks. Many of our speakers have kindly agreed to share their slides with you, and these are available within the literature section on the platform. Some of our speakers will have included their contact details in their presentation. If they do, please feel free to reach out to them directly. If you'd like to contact the online symposia team at any point, please contact us via the Technical Help tab at the bottom of the navigation bar on the left. Now, with that out of the way, I'd like to introduce the first talk of the day: Stem Cells and Genomics, from Precision Medicine to Clinical Trial in a Dish. I am pleased to welcome Joseph Wu as your presenter. Joseph Wu, MD, is Director of Stanford Cardiovascular Institute, and Simon H. Sturzer, MD, Professor of Medicine and Radiology at Stanford University. He is a board certified in cardiovascular medicine, and his lab works on cardiovascular genomics and induced pluripotent stem cells. The main goals are to understand basic disease mechanisms, accelerate drug discovery via clinical trial in a dish concept, and implement precision medicine for patients. He is listed as the top 1% of highly cited researchers by Web of Science for the past 5 years. A warm welcome to you, Joseph. Following the presentation, we'll have a Q&A session, and we'll welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Joseph's presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. Now, without further ado, I will hand over Joseph to begin his presentation.
So thank you, Carolyn, for the kind introduction and also invitation to the Innovations in Disease Modeling seminar. I think for the next 40 minutes, I'd like to give the audience here an overview on how we use the stem cells in genomics for precision medicine and clinical trials in a dish. Before I start, here's my disclosure statement. I think as you know, there's a significant interest in precision medicine in the US, and this concept was first promoted by President Obama back in 2015. Much of this is made possible by recent advances in next generation sequencing, bioinformatics, big data, and AI machine learning. By understanding our genes along with our lifestyles and environment, the hope is that we can better predict which treatments might work best for us. In my opinion, I think there are three key technologies that marries precision medicine and basic science research. The first breakthrough is DNA sequencing. And as you know, this technology has now become much cheaper with lower error rate, broader coverage, and much faster turnaround time. Back in 2000, it cost about $1 billion and over 13 years to get the DNA sequence. Today, you could get your DNA sequence for less than $1,000 and with turnaround time of about 3 weeks. So with increasing usage of next generation sequencing, there's hope that clinical care can be more predictive, preventive, personalized, and participatory. In my opinion, the second technological breakthrough is the various types of genome editing technologies, starting first with zinc finger nucleases, and then TALEN, and then culminating with CRISPR-Cas9 discovered by Emmanuel Charpentier and Jennifer Doudna in 2011. So using these genetic scissors, researchers can now change the DNA of animals, plants, and stem cells with extremely high precision. This technology has had a revolutionary impact on biomedical sciences. It is already contributing to new cancer therapies, and it may make the dream of curing inherited heart diseases come true in the future. In my opinion, the third key technology breakthrough is human-induced pluripotent stem cell platform, which was pioneered by Dr. Shinya Yamanaka. The ability to generate patient-specific and disease-specific iPS lines now provides you with a test bed to validate the human genetic variants using the CRISPR genome editing that I just mentioned. This then allows you to characterize the disease phenotype, identify molecular targets, and implement personalized medicine. Now, because of my training as a clinical cardiologist and the close working relationships that we've had with our clinical colleagues, over the past 10 years, our lab has generated iPS cells for more than 1,500 patients with different genetic background and cardiovascular diseases. And then from these diverse patient populations, we can then generate their patient-specific 2D micropattern human cardiomyocytes, 3D human cardiac organoids, as well as 3D human engineered heart tissues for disease modeling and also for drug screening. Now, with that as a background, I want to give the audience here some examples of how we use DNA sequencing, genome editing, and human iPS cells to answer key biological questions related to cardiovascular medicine. And so shown here is a schematic of different causes of heart diseases, which falls into three major buckets. The first bucket is primary cardiomyopathies, which could be genetic or acquired. Genetic cardiomyopathies would include, for example, hypertrophic cardiomyopathy, dilated cardiomyopathy, arrhythmogenic right ventricular dysplasia, left ventricular non-compaction, and others. Acquired cardiomyopathies could be inflammatory, such as COVID myocarditis. Next is secondary cardiomyopathies, which could be chemotherapy or radiation-induced cardiotoxicity. And then the third bucket is the more common causes that are more likely to be polygenic, such as coronary artery disease, hypertension, and congenital heart disease. And I'll show you several examples of what we've done in the past several years. One example is a familial hypertrophic cardiomyopathy, HCM. HCM affects about 1 in 500 people. HCM is one of the most common causes of sudden cardiac death in young adults. And then clinically, these patients have what we call left ventricular outflow tract obstruction, as well as cardiac arrhythmias. This is a diagnosis of exclusion when an athlete passes on the football field or the basketball court. And then to date, more than 500 mutations have been identified across more than 30 genes. About 12 years ago, we will refer a large family with familial hypertrophic cardiomyopathy by our heart failure cardiologist. The mother here had HCM with symptoms of cardiac arrhythmia. She has 8 kids. The 2 oldest already manifested the HCM phenotype, whereas kid number 3 and kid number 8 are both genotype positive, but phenotype negative, most likely because they were still young at that time. And then through a whole series of experiments, we were able to show that this particular myosin heavy chain 7 mutation with arginine to histidine switch at codon 663 causes increased intracellular calcium, which leads to calcium handling defect and then cellular arrhythmia. It also causes activation of the calcineurin NFAC signaling pathway, which leads to hypertrophic responses. And then importantly, treatment with calcium channel blockers such as verapamil and beta blockers such as propranolol can be used to reverse both the cellular hypertrophy as well as the cellular arrhythmia. So this is one of the earliest examples of how we use these patient-specific iPSC-derived cardiomyocytes as an in vitro platform for drug screening. Now, the example of hypertrophic cardiomyopathy that I just showed you earlier are considered classic Mendelian genetics with strong genotype-phenotype correlation. But as you know, human genetics is much more complicated and much more messy, especially when you're dealing with the variants of unknown significance, or VUS, which is really a clinical conundrum for clinicians taking care of these patients. For example, shown here is a program of a healthy middle-aged man who underwent DNA sequencing as part of an executive. Now, the example of hypertrophic cardiomyopathy that I just showed you earlier are considered a class of Mendelian genetics with strong genotype-phenotype correlation. But I think, as you know, human genetics is much more complicated, much more messy, especially when you're dealing with the variants of unknown significance, which is really a clinical conundrum for clinicians taking care of these patients. For example, shown here is the proban, who is a healthy middle-aged man who underwent DNA sequencing as part of an executive VIP workup. He was found to have a likely pathogenic variant in myosin light chain 3, with cysteine to alanine switch at codon 170, along with several members of his family, including his mother, his brother, and also his daughter. So the clinical dilemma is, what should we do with a healthy individual in which the DNA sequencing shows possible pathogenic VUS? So here, this case was referred to us, and we first generated iPS cells from family members with and without the VUS. Second, we showed that the cardiomyocytes and the VUS carriers were normal at baseline. Third, when we introduced a homozygous variant by CRISPR editing, meaning that the patient now has 2 copies of this potentially pathogenic variant, yet his cardiomyocytes remain normal. And then fourth, when we disrupted the healthy allele by introducing a frame shift mutation, the cardiomyocytes were also normal, meaning that what was annotated as a potentially pathogenic variant allele was still able to perform normal function. And finally, just to make sure that our assays were working properly, when we genome edited a well-known pathogenic variant of myosin light chain 3, his cardiomyocytes started showing severe arrhythmic phenotypes. So based on these 5 steps, and based on the fact that the patient himself was clinically healthy, we concluded that what was initially annotated as a likely pathogenic variant is most likely a benign variant. And because of this study, ClinVar, which is a commonly used genetic testing platform, has now included our findings in their annotation of this particular VUS as also possibly benign. Now, besides the 2 examples of a classic genetic mutation and VUS that I just showed you, we're also interested in studying the impact of single nucleotide polymorphism, or SNP. In this case, we focus on the SNP for aldehyde dehydrogenase, which is one of the most common genetic polymorphisms in the world. This enzyme deficiency is present in about 8% of the world population and 36% among East Asians. It causes an alcohol flush reaction, which include nausea, flushing, facial flushing, headache, and tachycardia. Antabuse or disulfiram works on similar mechanism and is used to help alcoholics stop drinking because they will feel lousy afterwards. Now, interestingly, the aldehyde dehydrogenase 2 polymorphism involving glutamic acid to lysine switch has been linked to increased risk of coronary artery disease, more severe outcomes after heart attack, and a higher incidence of hypertension and cancer, as well as increased complications of type 2 diabetes. This slide shows the enzyme deficiency has been well documented to be a risk factor for coronary artery disease and myocardial infarction in most East Asians. This piqued my interest in terms of studying this particular variant. And here you can see epidemiology studies for Japanese men, Korean men, and also Han Chinese. Now, to model the effect of this SNP on worse outcome after myocardial infarction, we recruited 10 Stanford undergrads who are of East Asian descent. 5 of them are wild type and 5 of them are heterozygous, which means they'll have symptoms such as headache, facial flushing, and tachycardia. Now, if you're homozygous mutant, which is about 1 to 2% among East Asians, the symptoms get even more severe. Now, here we modeled a myocardial infarction by exposing the iPS cell-derived cardiomyocytes to hypoxia. As you can see, the wild-type cardiomyocytes tolerated the hypoxia quite well with minimal apoptosis, whereas the heterozygous cardiomyocytes had significant apoptosis with hypoxia. Overall, we believe that the enzyme is involved not only in detoxifying alcohol, but also getting rid of the reactive oxygen species, or ROS. And after a heart attack, there will be tons of ROS that get activated. And if you're deficient in this enzyme activity, you have a harder time getting rid of ROS, and hence leading to more cell death, and perhaps explaining for the worst clinical outcome observed in epidemiology studies. Now, the last study I show you focuses on the effects of hypoxia on iPS cell-derived cardiomyocytes to model myocardial infarction. As a follow-up study, we recently used iPS cell-derived endothelial cells from these ALDH2 deficient patients and exposed them to alcohol to model vascular dysfunction and coronary artery disease. Now, we know from large genome-wide association studies using Biobank Japan dataset that the ALDH deficiency is one of the strongest SNPs associated with CAD. Mechanistically, this is because alcohol is broken into acetaldehyde, which is toxic and causes damage to the DNA, increases ROS, and increases vascular inflammation. Alcohol is further broken down into acetic acid by the aldehyde dehydrogenase enzyme. Acetic acid is non-toxic. And so if you're missing this particular enzyme, you will have more acetaldehyde in your systemic circulation, which is damaging to the vascular system. And then importantly, here we did a compound library screen and molecular docking analysis to show that SGLT2 inhibitor, empagliflozin, can actually mitigate the vascular dysfunction. Empagliflozin alleviates the ALDH oxidative stress and nitric oxide reduction by inhibiting the sodium hydrogen antiporter activity and also activating the AKT-ENOS pathway. These findings were also confirmed in mouse models of diabetes and alcohol exposure, showing that the empagliflozin can ameliorate this endothelial dysfunction. So this is just one example using race-specific iPS cells to study the mechanisms of alcohol flush variant among East Asians. In the next few years, undoubtedly, you'll see more and more examples of investigators using iPS cells from different ethnicities to study various biological questions. For example, from a health and science perspective, we're interested in understanding why African Americans have much more hypertension, why Hispanics have much more diabetic vasculopathy, why South Asians, for example, Indians and Pakistanians, have much more metabolic syndrome. So I want to switch gear now to the second part of my talk, which is using these human iPS cells for drug screening. This is a major focus of research in our lab. I think there are several reasons for developing human iPS cell-based models. And these include, as I showed you just previously, understanding the mechanisms of diseases, which will then help us identify molecular targets and develop new drugs that can ultimately improve human health. And as you know, this process is both time-consuming and expensive. For example, it typically costs about $1 to $2 billion and about 10 to 15 years to bring a new drug from drug discovery phase through clinical trial to market approval. Even worse, many of the drugs that you take today actually do not work at all. So shown here are the top 10 highest sales for drugs in the US. Blue means the drug works and red means the drug does not work. So for Abilify, which is used to treat schizophrenia, out of 5 people, the drug only helps 1 person. For Nexium, which is used to treat heartburn, out of 25 people, the drug only helps 1 person. So overall, this is pretty poor response rate, and this is the reason why the editorial here is titled A Time for One Person Clinical Trial. In particular, we have an interest in chemotherapy drugs, and that's because heart disease and cancer account for the top 2 causes of morbidity and mortality in the US. As more cancer patients survive due to better chemotherapy and immunotherapy, more patients will end up with heart failure symptoms due to cardiotoxicity from these drugs. This is also the reason why we see significant growth in cardio oncology clinics and programs over the past 10 years. As an example, shown here is a study we published about 7 years ago. Here we recruited 8 breast cancer patients, and a year later, some of them had normal cardiac function, while others had doxorubicin-induced cardiomyopathy. We generated patient-specific iPSCs-derived cardiomyocytes and exposed them to doxorubicin in vitro. We then studied cellular responses by contractility, calcium cycling, metabolism, gene expression, apoptosis, and chemoprotection assays. And here, as a proof principle, we showed that if we pick the patients appropriately, these iPSC cardiomyocytes can be used to recapitulate which patients are more susceptible to doxorubicin-induced cardiotoxicity. So this slide shows the patient profiles. We recruited 4 healthy individuals as control. We recruited 4 breast cancer patients who were treated with doxorubicin and did not develop any heart failure symptoms, which we call the DOX group. We also recruited 4 breast cancer patients who were treated with doxorubicin but developed cardiotoxicity, which we call the DOX-TOX group. Now, none of these patients had other cardiac conditions, such as previous myocardial infarction or valvular diseases or heart surgeries to confound the clinical presentation. So here, for example, you have a relatively young 40-year-old woman whose baseline left ventricular ejection fraction was about 77%, but then went down to about 36 to 50% after 4 cycles of doxorubicin treatment. So for one of the experimental designs, we exposed the cardiomyocytes from the DOX and the DOX-TOX patients to the same dose of doxorubicin. At 1 micromolar, you can see the DOX-TOX cardiomyocytes had much more sarcomere disarray, suggesting damage to the cellular architecture. And then at 1 micromolar, the DOX-TOX cardiomyocytes also had more cell death, more cellular arrhythmia, and also more cellular apoptosis. So this is quite interesting because even though we do not know the exact variants or SNPs that can account for the patient-specific differences in chemotherapy-induced cardiotoxicity, these iPS cells are now serving almost like a mini chip readout that incorporates all of the patient's genomics, SNPs, transcriptomics, and proteomics, and it's giving you a readout of what happens when the individual patient's cardiomyocytes are exposed to various concentrations of doxorubicin. Now, besides doxorubicin, other popular chemotherapy drugs include tyrosine kinase inhibitors, or TKIs. So these TKIs kill cancer cells by inhibiting the phosphorylation activity of hyperactive receptor tyrosine kinases. Common TKIs that you may have heard of include Gleevec and Sutent, which inhibit VEGFR2 and PDGFRB in cancer cells, but can also cause heart failure side effect. And this is because both the VEGF and PDGF are signaling pathways of critical regulators of cardiovascular development. So in this study, we generated iPS cells from 13 patients, treated them with 21 most common TKIs, and came up with a cardiac safety index that allows us to predict which TKI is cardiotoxic and which TKI is not. Our ultimate goal is to further optimize this cardiac safety index so that we can better predict the safety profiles of chemotherapy drugs before giving them to patients. Now, in the US, both the FDA and pharmaceutical companies Also thinking about using iPS cell-derived cardiomyocytes to accelerate the drug development process. They recently published a study in which these cardiomyocytes were used to predict arrhythmia risk as part of the FDA comprehensive in vitro pro-arrhythmia assay, or CIPA initiative. I think, as you may know, all the drugs need to be tested for cardiac arrhythmia before they can get approved by the FDA for human usage. And then currently, the platform is testing them on the Chinese hamster ovary cells or the human embryonic kidney cells that artificially overexpress the hERG potassium channel. This is not ideal because the CHO cells and the HEX cells are obviously not cardiomyocytes. And so here, the investigators used the one consensus protocol, 2 human iPS lines involving 3 continents, 28 blinded drugs, 2 statistical models, and 10 volunteer labs. And importantly, the investigators were able to show an 87% prediction rate, which is very good considering that this is one of the earliest studies by the FDA CIPA group with a significant room for improvement in the future. Now, as you can see from these examples I just showed you, we are integrating iPSC biology with various cellular, molecular, and functional readouts, and also multi-omics analysis to help us better understand the disease at a systems biology level. I think in the next few years, you'll see more and more examples of investigators using large-scale human iPSC population to study various differentiated cell types, such as heart, brain, liver, muscle, and lung, and also to study expression quantitative trait loci, or EQTL, and then also to study genotype-phenotype relationships via these co-expression networks. And in my opinion, the drug discovery application will advance significantly in the next decade. One main reason is because of the remarkable progress in the AI machine learning technologies. For example, working in collaboration with computational scientists with Greenstone Biosciences, we now routinely use AI machine learning softwares that allows us to analyze the protein structure by in silico molecular docking. Once we know the molecular target and the protein structure of the diseases that we are going after, we can then do virtual screening of about 1 billion compounds at a time. And once we come up with the drug hits, instead of testing them on mouse cells, we'll be testing them on patient iPS cells that have the exact diseases that we designed the drugs for. We think this is much more accurate and also much more clinically relevant because of mouse and humans, physiology-wise, they're not the same. And I think in case you're wondering, this video here shows 3 drugs that bind to the protein target that we're interested in. Here you can see that the drug in pink color binds to the target, whereas the other 2 drugs do not. And so the drug in pink color will be something that we will pursue instead of the other 2 drugs. So here I just want to highlight one example of using the AI machine learning and also in silico docking for drug discovery. I think, as you may know, marijuana is being used increasingly in the US due to legalization. Interestingly, the UK Biobank database shows that marijuana use is associated with increased myocardial infarction. However, the exact mechanism is unknown. And here we show that THC, which is the psychedelic component of marijuana, can cause vascular inflammation and also oxidative stress by using iPS cell-derived endothelial cells. We're confident that the mechanism is through binding of the THC to the cannabinoid 1 or CB1 receptor, which then activate the MAP kinase pathway, which then causes oxidative stress, inflammation, and cell death. And importantly, using in silico chemical library screen, as shown here, we found that genistein, which is an isoflavone, serves as a neutral CB1 antagonist that can competitively block the binding of the THC to the CB1 receptor, and hence block the harmful effect of the THC. These findings were also confirmed using iPS cell-derived endothelial cells in patients exposed to THC, showing that increased inflammation in blue, and then with genistein, which decreases the inflammation in red. And finally, the drug target hit for genistein was also confirmed in 2 independent mouse models of coronary and carotid atherosclerosis. So for the last part of my talk, I want to touch upon how we can then use human iPS cells to implement the concept of clinical trial in a dish. Here, the concept is that we should be able to recruit patients generate their differentiated cell types of interest, test which patients respond to drugs and which patients do not by using their iPS cells as surrogate, and then go back to perform the actual clinical trial on the patients who are positive responders to the drug. That way, you have a higher chance for success rate for your phase 1-2 clinical trials, decrease the cost, and also accelerate the timeline for drug development. In my opinion, this iPSL model is ideal for developing new drugs or repurposing existing drugs for rare orphan diseases. There are about 7,000 rare diseases known to exist today, yet there are only about 770 orphan drugs that have been approved by the FDA. The idea is that you can generate iPS cells from these rare orphan disease patients, test which drugs work and which do not, and then initiate your phase 1 clinical trial that way. As one example, in this first trial of drugs using iPS cells, Japanese investigators study patients with rare genetic disease called fibrodysplasia ossificans progressiva, or FOP, also known as Manchmal disease. This disease is caused by a mutation in the ACVR1, or activin receptor type 1 gene, in the bone morphogenic protein pathway. It is very rare, occurs in about 1 per million people. Affected individuals have a life expectancy of about 40 years. Patients die when their muscles and tendons are gradually replaced by bone. And because it is so rare, it would have not been possible to do a conventional clinical trial. And here, the Japanese investigators generated iPS cells from FOP patients. They screened for 6,800 drugs, identified rapamycin as an effective molecule for preventing bone formation, and have now initiated phase 1 clinical trial. Now at Stanford, I also want to tell you an example of clinical trial in DISH. About 6 years ago, we were referred by our electrophysiology colleague, a large family cohort with lamin AC frameshift mutation that leads to early termination. Lamin cardiomyopathy accounts for about 10% of familial dilated cardiomyopathy, so roughly 1 in 25,000 patients. And in this large family, many of the affected individuals had cardiomyopathy, atrial fibrillation, and atrial ventricular block, and 8 of them died from sudden cardiac death. And then using iPSL cardiomyocytes, we confirmed that the lamin mutation is indeed responsible for the cellular arrhythmia and impaired contractility. And as shown in this cartoon, typically the healthy control cardiomyocytes have normal expression of lamin protein, normal nucleus structure, and normal lamin-associated domain. However, patients with a lamin AC mutation have abnormal nucleus structure that causes increased open chromatin in lamin-associated domains instead. This then leads to activation of genes that should have been silenced in otherwise healthy conditions. And using RNA-seq and ATAC-seq, we identified PDGF signaling pathway as one of those abnormally activated genes in these lamin mutation cardiomyocytes compared to the control cardiomyocytes. Now, what's interesting is that we noticed many of these lamin patients within this family developed early onset hypertension at age 30s and 40s. And here, we differentiated their iPS cells into endothelial cells, which then allows us to compare the differences between the lamin patients and the healthy controls. We perform a whole battery of functional assays and identify KLF2 as a potential transcription factor that is responsible for the endothelial dysfunction. And then using FDA-approved drug library screen, we found that lovastatin, which is a potent KLF2 agonist, can be used to rescue the endothelial dysfunction. We now want to test to see if this is true in the actual patients themselves. So the same lamin patients whom we tested their iPS cell endothelial cells were then asked to return to our clinic, and we treated with lovastatin daily. The clinical data is shown here. We use EndoPAT, which is a machine that can assess flow-mediated dilatation during reactive hyperemia. Normal index is considered 1.67 and above. And as you can see here, lamin patient number 2 started with a poor index of 0.48 and improved significantly to 1.06 after 6 months of lovastatin treatment. So just like the example I showed you earlier with the Japanese investigators using iPSL for FOP patients to identify rapamycin as a drug hit, I think this is another example of a clinical trial in a dish, whereby we first identify a large family with lamin mutation. We then screened for a panel of FDA-approved drugs using their iPS cells and found that lovastatin can be used to improve their cellular phenotype by enhancing the KRF2 pathway. We then confirm these in vitro findings in the actual patients themselves by using EndoPath to show functional improvement after 6 months of a lovastatin treatment. So to conclude, I just want to highlight the Stanford CVR iPSR Biobank. So far, we have generated about 1,500 lines from patients with different cardiovascular history, ethnicity, and sex. In collaboration with the NHLBI TIMET program, we're in the process of finishing DNA sequencing all these lines. We also use PharmGK to help us understand how human genetic variations impact the drug response phenotypes. We have established a robust sharing resource plan with many investigators so that if you're interested in some of these lines, please feel free to contact me and we'll be happy to send them to you. We're working with the NIH on biobanking and also open sharing resource. Lastly, we're working with the FDA on drug safety testing. And I think this last aspect of working with FDA is very important if we want to use this platform for accelerating drug discovery. As some of you may know, the FDA Modernization Act 2.0 was recently passed by both the Senate and House on December 2022. The law authorizes the FDA to use alternative testing platforms, such as stem cells, organoids, and in silico modeling, and for some drugs to completely eliminate the requirement for animal testing before entering clinical trials. So the goal here is to improve the predictiveness of preclinical safety and efficacy assessment, and also to reduce the use of animals in drug discovery and development programs. Now, this slide is given to me by my colleagues at the FDA. I think for rare orphan diseases, as I explained earlier, the FDA recognizes the challenges of developing new drugs due to small number of patients, and yet there are thousands of genetic variants for each disease. So now their approach is to accept data generated from cell models to test a drug efficacy instead. As an example, for cystic fibrosis, the drug Callidical made by Vertex Pharmaceuticals was initially approved for 10 genetic variants and now has been expanded to 24 more variants. Another example is Fabry's disease, which is a lysosomal storage disease, which is passed on via the X chromosome. The drug Galafold made by Amicus Therapeutics was initially approved for 40 genetic variants and now has been expanded to 348 genetic variants based on cell models. And currently, the FDA is interested in expanding the cell-based platform across a full spectrum of the diseases. This slide is also given to me by the FDA. I think the value proposition here is that using the in vitro efficacy data to reduce the number of clinical studies, to inform regulatory decision-making at the time of marketing application, to streamline drug development, and also to expand the indications based on the in vitro data. So in summary, I believe the paradigm shift in the future for drug discovery will involve patient-specific iPS cells that allows us to understand the molecular mechanism of diseases. These iPS cells can then be used to differentiate the different tissues, such as patients' heart, brain, liver, kidney, and muscle. Along with clinical genomics, this then provides us with disease-specific and tissue-specific gene expression data sets, which then allows us to use AI machine learning for molecular docking, molecular dynamic simulations, and also in silico ADME predictions. And then by combining iPS cell genomics and AI machine learning, we believe that you'll give us drug targets that are much more precise and predictive. And finally, the lead compound can then be tested on iPS cells that have the desired disease phenotypes that you designed the drugs for, hence completing the loop for a clinical trial in a dish. This concludes my talk. I want to acknowledge my postdocs, graduate students, instructors, my collaborators, and also my former trainees whose work I highlighted in my talk. I also want to thank funding support from the NIH, from the AHA, Chan Zuckerberg, FDA, and also collaboration with scientists from the startup company Greenstone Biosciences. Finally, I would like to encourage all of you to attend the 2023 Stanford Drug Discovery Symposium. This is both an in-person and virtual meeting because we have strong corporate support. The registration is free for this meeting. We'll have talks by the CEOs of Genentech, Gilead, Amgen, and Illumina. We'll also have talks by the CSO and CMO of Big Pharma. We'll also have talks by the FDA Commissioner, Rob Califf, as well as FDA Directors of Center for Biologic Evaluation, Peter Marks, and also Center for Drug Evaluation, Patricia Cabazzoni. There will be a Nobel Prize session in which we have fireside chat with 4 Nobel Prize winners, Carolyn Bertozzi, Jennifer Doudna, Brian Kabilka, and Roger Kornberg, on how their Nobel Prize discoveries have led to new treatments in small molecules, antibodies, drugs, genes, and cell therapies. So I encourage all of you to attend either in person or virtually. Thank you very much for your attention, and I'll be happy to take any questions.
Okay, so that was an incredible talk, and I can see that we've received some questions already, so I'll move straight on to the Q&A now. So your first question is, what similar are the iPSC-derived cardiomyocytes compared to the human cardiomyocytes?
Yeah, so that's a very good question, Carolyn. I think the human iPSC-derived cardiomyocytes, they are considered at the fetal stage compared to the human adult cardiomyocytes that you can biopsy from patients. Now, this goes the same for other types of iPS lineages. For example, the iPSC-derived brain cells or iPSC-derived liver cells or the iPSC-derived skeletal muscle cells. They will be considered more fetal stage compared to the adult cell types. So this is one of the major limitations of the 2D in vitro differentiation techniques. And I think for that reason, many groups, including our group, are constantly trying to upgrade our differentiation protocols to make these iPSC cardiomyocytes more mature. And so some of the techniques that the investigators have used, for example, include electrical stimulation, mechanical stretch of these cardiomyocytes. You can also change the substrate stiffness where the cells grow. You can do genetic manipulation by overexpressing or inhibiting some of the key developmental genes involved in cardiac differentiation. You could also add biochemical factors such as fatty acids and thyroid hormones to induce maturation. And finally, many groups are co-culturing these cells with other cell types to generate a 3D organoid or 3D engineered heart tissues. So this is overall a very active area of investigation these days.
That's great. Thank you very much, Joseph. And we've got time for a couple of more questions. And your second question is, for drug screening, what is the typical process involving primary, secondary, and tertiary screen?
Yeah, so that's also a very good question. I think for the drug screening that we do in our lab, we typically use the 2D iPSC cardiomyocytes or 2D iPSC endothelial cells that I show you. The exact cell type would depend on the drug that we're designing for. For example, drug targeting cardiac metabolism will use cardiomyocytes. Drug targeting antifibrosis will use the cardiac fibroblasts. And the reason why we use 2D monolayer culture as primary screen is that it's relatively low cost and high throughput. And then this allows us to screen thousands of compounds at a time. Now, once we get some hits, for example, in the range of about 10 to 20 compound hits, then we can go to the secondary screen, which will involve more cell lines, typically about 10 to 20 cell lines at a time. This allows us to first confirm that our data is true with a bigger sample size. And also, it also allows us to understand the variability of drug responses that happens across different individuals, which is perhaps the most important question. Now, once we narrow that list of drugs from 10 to 20 to about top 2 to 3 hits, we will then go to our tertiary screen. And this tertiary screen typically involves using 3D engineered heart tissues or 3D cardiac organoids. The reason is that this allows us to tease out the effect of the drug in the context of all the key players, all the key cell types in the heart, and the crosstalk that occurs among these different cell types. And then finally, once we confirm the lead compound in these primary, secondary, and tertiary screens, we would then proceed to animal studies looking at safety and efficacy, and also PK/PD or pharmacokinetics and pharmacodynamics of this lead compound. So this is a general summary of how we go about with our primary, secondary, and tertiary screens.
That's fantastic. Thank you, Joseph. And we've got time for one more question. What is the biggest hurdle for implementing clinical trial in a dish?
Yeah, so I think for most academic labs, the biggest hurdle of implementing this clinical trial in a dish concept, in my opinion, are time and cost. It is actually quite labor intensive for a single postdoc to handle multiple IPS lines, and it can be quite expensive to culture many plates at a time because of the large amount of cell culture media and reagents that are needed. I think for industry, this is probably less of an issue given that they have much more resources than most academic labs. And so one way to address this time and cost issue, in my opinion, is to use this quote unquote cell village concept. This concept involves culturing multiple cell lines at a time by taking advantage of the single cell RNA sequencing technology, which allows you to identify the gene expression at a single cell level. And as long as you also have the DNA sequencing of these multiple cell lines, you can then deconvolute to see which data point comes from which patient. And I think this approach will be more widely adopted in the future and can help address this bottleneck of time and cost for clinical trials in DISH, especially for most academic labs.
That is great. Thank you, Joseph. Unfortunately, that's all we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will be made available to view whenever you want for the next 30 days. So that just leaves me to say thanks again, Joseph, and we will be back soon with the next talk.
Thank you very much.